Picture a misty New Zealand forest where a bird towers like two adults. The giant moa vanished, but could return.
Filmmaker Peter Jackson poured millions into reviving this giant. Why support gene editing to raise a lost bird?
Scientists will rebuild moa genes from ancient bones, then edit a small bird to carry those traits. Is this science fiction?
Stay with us as we unpack the science, culture, and hope of reviving this lost giant. Ready for the journey?
Welcome back, Pack. After that cliffhanger, let's dive into the heart of the giant moa story.
Yes, our voices are synthesized, but we're real conservation storytellers who've spent countless hours with scientists and elders.
This episode explores the extinct giant moa, the science behind de-extinction, and the cultural questions around it.
We start with history, then dig into gene editing, debate, show real experiments, feature your comments, and end with future visions.
We want your voice too. Drop questions, vote in polls, and share your own conservation dreams.
Pause and open the sources panel as we go. Trust and curiosity are the backbone of this channel.
The giant moa was an enormous flightless bird from Aotearoa, towering above shrubs and weighing as much as a young cow. These browsing herbivores evolved in isolation, but vanished about 600 years ago when Polynesian settlers arrived and hunted them. For Māori, moa bones and stories are taonga. Elders still speak of their feathers and drum-like calls.
Ethnobiologists remind us that bringing back a species is not just biology. It touches culture, land, and rights.
Filmmaker and bone collector Peter Jackson grew up exploring fossil sites and dreamed of seeing these birds walk again. In July, he and Fran Walsh invested millions into a de-extinction startup to pursue that dream. Jackson said, "This project isn't about playing God. It's about inspiring hope and helping living species."
Scientists caution that without cells or eggs, we can't clone a moa. We can only edit living birds to carry traits.
Researchers started by sequencing ancient moa bones to map their DNA. The Little Bush moa genome was a breakthrough.
Geneticists found moas are closer to tinamous than kiwis, so a small ground bird may be our genetic canvas.
CRISPR lets scientists cut and paste DNA. They hope to edit thousands of tinamou genes to resemble a moa.
It's not simple. Moas diverged from tinamous tens of millions of years ago, so thousands of changes may be needed.
Even with advanced gene editing, experts estimate a 5 to 10 year timeline before we see a chick hatching.
Bird reproduction is tricky. We can't transfer embryos, so we need to transplant edited germ cells into surrogate eggs.
The team edited wolves to express dire wolf genes and mice with woolly hair. They see these as stepping stones.
Those animals are hybrids, not true de-extinctions. Experts remind us they are proxies, not resurrected species.
Milestones include sequencing the moa genome, raising dire wolf pups, launching the project, and restoring native habitat.
Environmental groups are removing predators and replanting forests to prepare for the possible return of large herbivores.
Ngāi Tahu researchers guide ethics. Some elders see benefits. Others ask whether a lab-made bird counts as taonga.
Ecologists worry that a 500-pound browser could damage modern forests and outcompete native birds and plants.
Supporters argue that gene editing tools could rescue endangered species by restoring lost genetic diversity.
Critics fear that chasing extinct giants diverts money and attention from saving the species that still survive.
Jackson says this project is a rallying cry for conservation, not nostalgia. He wants to inspire action for living species.
Skeptics respond that nature isn't a movie set. We can't script ecological outcomes with editing tools.
Through this story, you'll see how science, culture, and ethics collide. Keep an open mind and heart.
We've outlined the moa's history, extinction, cultural ties, and basic plan. Next, we'll dive deeper into the science.
Are we brave enough to alter a living species for the sake of one we lost? Let's find out.
As we move forward, remember that extinction is forever. Even with proxies, we can't turn back time.
Share your initial feelings in the comments, awe, worry, or excitement, and we'll revisit them in our debate.
Ready to trade emotion for data? Next, I'll walk you through genomes, gene edits, and numbers. Let's tackle some myths right away. This isn't Jurassic Park with instant dinosaurs, but careful gene editing in modern labs. De-extinction here means editing a living genome to express traits of an extinct cousin, not regenerating a whole genome from nothing. Headlines about bringing back the MOA refer to a bird with MOA-like features created by thousands of DNA edits.
We're talking about a mosaic, not a clone. That difference matters for our expectations and ethics.
Ancient DNA is fragile. Scientists recovered fragments from bones and pieced them together like a puzzle. The Little Bush MOA genome provided a blueprint of many DNA letters, revealing genes for growth, immunity, and feathers. Comparing MOA and tinamou DNA shows many differences are small switches controlling bone length and metabolism. Scientists use CRISPR gene scissors that cut DNA at specific spots and paste in MOA versions. Editing thousands of sites raises off-target risks, and each change can affect development or behavior.
If we push a tinamou embryo too far, we might make a sick bird. Careful testing in cell cultures is needed.
Another myth is that we already have a MOA cell to copy. In reality, we only have DNA sequences. That means cloning, like we did for Dolly, isn't possible. We need living eggs from a related species. For birds, scientists edit germ cells in a lab and insert them into surrogate embryos to grow chimeric mothers. Those mothers will lay eggs with edited germ cells, producing chicks with a mix of donor and host DNA. Proof of concept experiments use chickens and pigeons as surrogates for dodo and auk projects. Results are pending.
That's a lot of steps. Edit, implant, hatch. Each has to work before we see a MOA-like chick.
Scientists also have to program growth hormones and bone patterning genes to recreate the MOA's towering height and mass. Some growth genes also affect brain development, so unintended changes could cause complex behavioral issues.
It's not as simple as turning on a single giant gene. Thousands of interactions shape a living animal.
Thinking a proxy solves conservation is wrong. If the habitat isn't ready, animals may fail or disrupt ecosystems.
Ecologists note that invasive mammals abound. A giant browser could spread seeds, but also topple saplings and nests.
Predators and deer alter the landscape. Adding a MOA proxy might tip the balance, harming kiwi and other birds.
Conservationists argue that resources could instead support endangered kiwi and kakapo and fund forest restoration work right now.
Ecologist Philip Seddon says, extinction is forever. Proxies are hybrids, so we shouldn't blur that line.
Proponents counter that we already modify endangered species genomes to fix disease or restore variation. This is a spectrum.
Scientists edited a gray wolf embryo to include a dire wolf gene, producing a pup with different growth traits. They also inserted a mammoth hair gene into mice, yielding fluffy coats. These illustrate that single trait edits are possible. A MOA proxy would require editing thousands of genes across growth, metabolism, and immunity. It's far more complex. Gene editing is accelerating. CRISPR is becoming more precise, and new base editors can swap letters without cutting. Still, the bigger challenge is scaling. Editing a handful of embryos is one thing. Producing a healthy flock is another. There's also an ethical question. If we create these birds, who owns them? Researchers, investors, or iwi? Ngāi Tahu leaders ask whether a lab-made MOA counts as taonga, a sacred treasure, or just another commodity. Some Māori see ecotourism benefits. Others fear commodification and prefer focusing on living species restoration. Cultural consultation is ongoing. The project includes scholars and elders to ensure respect, but consensus is far from reached. Another myth is that the MOA's environment is unchanged. In reality, New Zealand's ecosystems have shifted. Large herbivores shape forests. Without them, some plants grew bushier. Reintroducing one could change ecosystems. Biologists plan to model effects by simulating browsing and dispersal to predict whether a proxy would help or harm.
Removing stoats, goats, and deer could make room for a browsing bird again. This helps regardless of de-extinction.
Some think we can just enlarge a chicken. Body size is controlled by many genes and hormones, not a single switch. A MOA's long legs and neck require altered bones and blood pressure systems, which evolved over millions of years.
In gene editing experiments, animals often end up smaller or fragile. The path to a robust MOA-like bird is uncertain.
Funding comes from private investors like Jackson, not taxpayers. But every dollar has opportunity costs.
Viewers have asked whether that money could protect kakapo or kiwi instead. We'll ask you again in our debate section.
When hearing promises, ask for evidence. Are timelines realistic? Do pilot studies exist? That's how we cut hype.
We've debunked myths and laid out the steps. Keep those questions in mind, because our debate section starts now. I can't help but feel a mix of wonder and responsibility. The thought of a towering bird walking through podocarp forests again sparks awe. Yet my heart worries about how it would fare in a world crowded with deer, stoats, and humans. In our comments, some of you wrote that resurrecting giants is like inviting an invasive species. Others said you dream of hearing a drum-like call echo through the valleys. Which side of your heart speaks louder?
I hear that tension. As scientists, we have to weigh hope against data. We have a genome blueprint, but not a living cell. We know how to edit thousands of genes, but not how those edits combine in a real bird. Every dollar invested here is a dollar not protecting kiwi or restoring wetlands. So I ask, how do you weigh a symbolic act against immediate needs? Would you reallocate funds, or is the inspiration worth it?
Beyond cost, this project sits at the intersection of science and culture. Māori scholars emphasize that any attempt to revive taonga must be led by iwi, not outsiders. Some elders worry a lab-made MOA might feel like appropriation, while others see it as a way to celebrate and share heritage. Who should decide whether to go ahead? Should economic benefits from ecotourism matter, or is cultural consent paramount?
There is also ecology. A MOA proxy would likely weigh hundreds of pounds and browse on shrubs all day. New Zealand's forests have evolved under centuries of browsing by deer and goats. Some plants may be ready, others not. Without predators, these birds might overpopulate or spread seeds of invasive plants. Our models can't predict everything. Are we prepared to risk an experiment on the landscape if it inspires a generation?
On the flip side, every breakthrough here could help living species. Perfecting germ cell transplants could rescue the kakapo, red wolf, or Spix's macaw. When we learn to control growth genes, we might fix developmental disorders in endangered animals. If Peter Jackson's blockbuster investment funds that research, maybe the MOA is just the trailer for a feature on saving those still with us. That twist makes me smile. Does it change your view?
So we circle back. This is neither a simple monster movie fantasy nor a villainous distraction. It's a complex research program with potential upsides and real risks. I remain skeptical of the timeline and scope, but I respect the effort to innovate. Tell us in the comments, if you had to choose, would you invest in gene editing for de-extinction, habitat restoration for existing species, or both equally? We'll run a poll and discuss your responses.
And while you ponder, share your pun of the week. Our community is known for mixing science with smiles. Last time, someone wrote, "You can't spell determined without ermine" when we covered states. It made my day. Maybe you have a MOA pun waiting. I'll wait. After this pause, we'll explore real-world case studies, from dire wolf pups to woolly mice. Don't go anywhere.
Before we move on, open the sources panel for details on the studies we've cited. Check out the Little Bush MOA genome paper and the critique by Philip Seddon. Evidence matters. Take a moment to breathe and reflect. When you're ready, we'll dive into case studies that illustrate what gene editing can and can't do.
Let's start with Colossal's canine experiment. You might have seen headlines about a dire wolf being born in a modern lab. What actually happened is that researchers edited a single growth gene from the extinct dire wolf into a gray wolf embryo. That embryo was carried by a surrogate dog. When the pup was born, he looked like a normal wolf, except for slightly larger paws and thicker bones. There was no saber-tooth or monstrous hybrid, just a subtle shift. This proof of concept shows that single trait edits are possible, but also that changing one gene rarely transforms an animal into its extinct ancestor.
Another widely shared story involves mice with woolly coats. Scientists inserted a woolly mammoth hair gene into lab mouse embryos to see if they could produce thicker fur. The result? Tiny mice with slightly longer, denser hair, not shaggy mini mammoths. Why try this? Because fur texture genes are easier to study than complex traits like size. The experiment revealed that the inserted gene was active, but didn't disrupt other systems. It offers hope that editing structural proteins might be safer than editing hormones. Yet it also highlights how incremental these changes are. We're nowhere near reconstructing an extinct species.
A landmark moment for de-extinction came when researchers sequenced the genome of the little bush moa, a smaller relative of the giant moa. Sequencing ancient DNA from eggshell and bone fragments, they assembled most of the moa's genetic code and compared it to living ratites. The analysis showed moas are more closely related to tinamous than to kiwis, overturning assumptions. It also identified genes controlling bone growth and metabolism that could be targets for editing. Without this blueprint, editing would be guesswork. Sequencing efforts like this underpin all de-extinction projects, yet they get far less media attention than flashy experiments. When Jackson and his partner announced their investment, they partnered with the Ngāi Tahu Research Centre to ensure indigenous leadership. Their plan isn't just about making a bird, it includes habitat work. Conservation groups are removing stoats and goats from sanctuaries and planting podocarp and beech trees so large browsers can once again shape the forest. It's a reminder that bringing back a species without preparing an ecosystem would be unfair and doomed to fail. These restoration projects are valuable, even if a MOA proxy never hatches, because they help endangered kakapo and kiwi right now.
Gene editing isn't just about resurrecting ghosts. Conservation biologists are exploring how to use these tools to help endangered species today. Take the red wolf, a critically endangered canid in North America. Its gene pool is so small that some pups are born with congenital disorders. Researchers are sequencing genomes and planning to introduce healthy versions of disease genes using CRISPR. In New Zealand, scientists have sequenced the entire kakapo population, revealing inbreeding risks that could be alleviated with gene editing. These projects share techniques with de-extinction, but focus on living animals. They show how technological advances sparked by the MOA project could have immediate impact.
The common thread in these case studies is collaboration and transparency. The dire wolf experiment involved wildlife vets, geneticists, and ethicists. The woolly mouse project was openly published for peer review. The little bush moa genome was sequenced with input from Ngāi Tahu. Habitat restoration is led by community volunteers, and gene editing for endangered species will only succeed if it earns public trust. That's why we host these conversations. By staying engaged, you're part of a global dialogue about where science goes next. Ready to shine a light on our own community? The spotlight is coming up. I want to start by thanking Jasmine for saying our Dodo episode was excellent. Your feedback fills our hearts. You asked if the germ cell breakdown or the ethics debate resonated more. Let me know in the comments which part stuck with you. Your answers help us tailor these deep dives and keep them clear.
We also heard from a viewer who criticized the giant MOA budget and demanded scientists be fired. Remember, the $15 million investment is private money. It covers genome assembly, CRISPR research, surrogate breeding, and animal care, all under Māori leadership. If you had control of that pot, would you spend it on DNA work, surrogate breeding, or protecting living species?
Musketeer Chris, You continue to make us smile. You joked that you can't spell determined without ermine, and you asked whether dog translators work on wolf howls. We explained that our models are trained on domestic dogs and should abstain on wolves, and we're planning a comparison between wolves, coyotes, and huskies. Vote below on which you want to see first.
Mike Boing accused our deep sea special of being faked with AI and reminded us that ghost crabs exist. Fair question. All bioluminescent footage in that video came from CSIRO research. Whenever we use AI enhancements, we label them. Would you like more behind-the-scenes scientist clips or raw field footage? Your trust matters to us. Thanks for speaking up.
Richard from Victoria pointed out that most thylacine samples come from the TIG-RR lab in Melbourne and felt we ignored his emails about five-toed prints. We hear you. Specimen provenance and accurate tracks are vital in de-extinction work. Would you like deeper coverage of specimen collection or of genome assembly in our upcoming thylacine update? Choose in the comments.
These conversations remind us that science isn't just in the lab. It thrives in community. Your puns lighten our day, your questions sharpen our focus, and your critiques keep us honest. Keep them coming. Comment, like, and subscribe, and share this with someone who loves wildlife. Next up, we'll peer into the future of de-extinction and innovation. Looking forward, gene editing is becoming more precise. Base editors can swap individual DNA letters without cutting. Prime editing rewrites sequences like a word processor. Scientists are working on germ cell transfer techniques for birds that could one day allow chickens to hatch kiwi or even MOA proxies. These innovations could rescue endangered birds and perhaps one day bring back traits from lost species. Synthetic biologists are also exploring artificial wombs for birds, lab vessels that mimic egg development, so edited embryos can grow outside a surrogate. If that sounds wild, remember that mammalian organoids started as science fiction too, but these advances raise new ethical questions. Who regulates lab-grown life? How do we ensure welfare for engineered animals? We need clear guidelines before scaling up. Technological progress must be paired with consent and co-governance. Indigenous communities like Ngāi Tahu are setting an example by embedding cultural advisors in the labs. In our future, we want gene editing projects that respect cultural values, restore ecosystems, and benefit living species. That is the path to responsible innovation, one that lifts us all. Finally, let's remember that the goal isn't to play God, it's to heal what we've broken. Whether we bring back a MOA proxy or not, the journey can yield tools for conservation, medicine, and our understanding of life. Imagine a world where we prevent extinctions instead of reversing them. Keep that vision in mind as we head to our closing takeaways. De-extinction isn't magic. It produces proxies by editing living genomes. That's messy and uncertain. Yet the work sparks hope, advances tools, and inspires us to care for what remains. Use that spark to act. Support conservation, stay curious, and question hype. If this video moved you, share it. Comment with your thoughts, vote in our poll, and join the pack. Subscribe for our next episode on animal communication breakthroughs. Open the sources panel to explore the science yourself. Until next time, stay wild and stay kind.